Thank you very much, Annabelle. Thanks everyone for coming. Can I start out with a show of hands? How many people are students, undergrads or graduate students? Oh, good, lots. That's great. Whenever I prepare a talk, I always assume there are going to be lots of students in the audience, so I hope I've hit the right target. There's the title of my talk. I'll try to explain what that means over the course of the talk. I work at the Genelia Research Campus, as Annabelle mentioned. This is a very special place. I could spend an hour just telling you about Genelia, but I won't. Let me just say that these are the people in my lab who do the work. There's actually, you'll see two of them listed here are lab coordinators and two are former postdocs. So we have 6 postdocs in the lab. And we have small labs at Genelia, but we get a lot done in large part because we have this tremendous resource called the shared resources. And every postdoc in my lab benefits tremendously from help that they get various people in the shared resources. So really when I give a talk, it's not just my lab, it's my lab plus all these other folks who help us get stuff done. Okay, I'm going to talk about brain function today and deconstructing how memory works at the cellular level. Don't get your hopes up. I don't have all the answers. I'm just going to tell you a few things that we've learned along the way. And I'm going to break the talk up into 3 parts. You guys are guinea pigs. I've never done this talk before. In the first part, I'm just going to tell you, what the hell are we doing? What are we trying to accomplish? The second part, I'm going to talk about some work that pertains to formation of memories. And in the third part, which is also labeled number 2, about the use of memories. That's what happens when you make slides late at night, the night before the seminar. Okay, so what are we doing? I'm going to show you this video to try to give you an introduction to what we're doing. I shot this video with my iPhone yesterday at my in-laws' house. My in-laws recently moved from Texas to Virginia. You'll recognize the strong Texas accents. And this squirrel is on a squirrel-proof bird feeder. And it's squirrel-proof because the bar that the birds sit on is spring-loaded. And if a squirrel stands on that bar, it closes its access to the birdseed. So it's squirrel-proof. Here we go. Here we go. So this guy's got it all figured out. So I think this video is pretty cool just because it illustrates that animals are really intelligent. When they figure out what they need to survive, they're going to make it happen one way or another. And I think as neuroscientists, what we want to understand is how the brain and the body working together allow animals to do such incredible things. Not just physical feats like jumping up onto the feeder, but also the cognitive feat of realizing that it has to hang from its back legs so it doesn't put too much weight on the bar. And there are many approaches that people are taking to try to understand intelligence. Of course, there's a whole field of artificial intelligence and bioengineering, where people are trying to engineer intelligent robots, for example. There are systems neuroscientists and cellular neuroscientists who are interested in understanding how the brain works. And for me personally, I really would find it very satisfying if we could achieve an understanding of the brain at all these different levels. So, what is the relationship between artificial intelligence and real intelligence? How do real brains actually produce intelligence and produce the complex behaviors that emerge from intelligence? And what are the properties of the cells that make all of this possible, as well as the connections between the cells? And this is a very traditional way of thinking about understanding complex systems in biology. is to break it down into the cellular machinery that that implements complex functions, and to try to understand the system in terms of its cellular composition. So that is very much my goal as a neuroscientist, and I think it's maybe not the same goal that everyone in neuroscience has, but I wanted to start out by telling you where I come from. How many people in the room know who this is? Wow, we've got only a couple. That's great. Okay, this is Lee Sedol. Lee Sedol is the 18-time world champion at the game of Go, which is an ancient Chinese game, said to be the most complex game in the world. And there are far many possible iterations of moves in a game of Go than there are atoms in the universe. And therefore, it's impossible for a computer to compute all possible moves in any given situation. And therefore, it's much more difficult for artificial intelligence to actually get good at playing this game. So a couple of years ago, Google DeepMind developed this artificially intelligent system called AlphaGo, and they pitted it against the top professionals, ultimately having it play against Lee Sedol. And there's a Netflix movie about this. I highly recommend that you all watch it. It's an amazing account of the competition between AlphaGo and Lee Sedol. AlphaGo won the match 4-1, and it was emotionally shattering to Lee Sedol, and really to tens of millions of people who watched this happen in real time. The notion that a computer could beat a human at something that required so much deep intuition formed over the course of a lifetime was really shocking to people, and I think that the movie conveys that really nicely. AlphaGo's prominence at the top did not last long, however, because about a year later, the same group, Google DeepMind, announced AlphaGo Zero, which instead of learning from observing humans playing Go games, learned by playing against itself, and therefore with really little or no human knowledge of humans playing the games, this AlphaGo Zero was able to learn the game from scratch, and AlphaGo Zero was pitted against AlphaGo, and it won 100-0. So there is now really no chance that a human is likely to beat an artificially intelligent system at the game of Go anymore. So that's done. Why do I bring all this up? I bring it up because it raises an interesting question, which is, if we can create intelligence in a computer, does that mean that we understand it? I mean, is that enough for us to say, okay, now we understand intelligence? I would say, I saw someone shaking her head over here. I would agree, absolutely not. We don't understand it. Even the people who made AlphaGo and AlphaGo Zero don't really understand it. Now, they probably have some really valuable intuition into how these kinds of systems, these deep artificial neural networks function, and therefore they may have some intuition that would be really valuable for us to tap into as neuroscientists in terms of trying to understand how these complex systems actually function or may function in a real brain. But there are still things that real brains can do that artificial intelligence systems cannot do. And it's a very interesting question why it is that the human brain, for example, is able to learn things much more quickly. Even though these systems are very intelligent, they require an enormous amount of training that happens in fast computers for very long periods of time. And these are things that humans can actually learn, things that humans can learn quite quickly. Like small children can identify the difference between a dog and a cat easily, but artificial intelligence systems have to be trained with millions of pictures of dogs and cats before they can identify them reliably. So here's what an artificial neural network looks like. It's typically layers and blocks of elements, which sometimes people call neurons. And these neurons are very simple units that get input from other neurons. They sum them linearly to some threshold, and if they surpass some threshold of input, then they produce output to the next level. And by implementing rules about how the weights of the connections between these neurons change in these artificial neural networks, this is sufficient actually to produce artificial intelligence. Real neurons are really not like that. So this is kind of a cartoon diagram of a neuron in an artificially intelligent system. It's just an element that gets input and an element that produces output. Probably those of you who study neuroscience recognize this as something like a cell body and a synapse. And real neurons are no more like this than this is like a real human, okay? All of you are very familiar probably with what real neurons look like. These are 3 neurons that I studied during my PhD about more than 30 years ago now. And at the time, I became absolutely fascinated with the structure of the dendritic tree of neurons and why it is that they have such an elaborate structure and why cells in different parts of the brain, these are all 3 different cell types in different parts of the hippocampus, have such differences in their structure. And this was my view of how neurons work, what they looked like for a long time. Of course, they also have an axon that goes somewhere, but I didn't really think too much about that. And then along came the mouse light project at Janelia, where the idea was to trace out the axon arborizations of individual neurons with cellular resolution throughout the entire mouse brain. And I'm going to show you this movie of 3 neurons in the somatomotor cortex of the mouse, just because I think this illustrates how incredibly complex individual neurons are, even in a mouse brain. So these 3 neurons each have about 40 centimeters plus of axon associated with one cell body. So imagine one nucleus has to produce all the machinery to provide the proteins to these axons to support the function of thousands, probably tens or hundreds of thousands of synapses. These are now thalamocortical neurons that you're looking at. And there's some neurons in the cortex that descend into the pyramidal tract. These neurons are incredibly complicated. So not only are the dendrites complicated, the axons are complicated. And I think we all should kind of open our minds to what neurons are capable of in animal nervous systems. They're doing something that's much, much more complicated than the simple linear integrate and fire operation that's typical of neurons in artificial systems. Okay, so where am I going with all this? In my lab, we study the hippocampus because the hippocampus is such an important structure in memory. As I told you, we want to understand the cellular basis of hippocampal function and its contribution to memory. There's a lot of evidence. What just happened? There's a lot of evidence in both rodents. Come on. In rodents and humans, that the hippocampus is an important contributor to memory. These are some neurons in the subiculum of the hippocampus, which is a brain region I'll talk about more extensively in a moment. They also have very diverse and very complicated elaborations of their axon arborizations, targeting many different parts of the brain. And one point I want to make here is that there's a lot of diversity in the cell types in the hippocampus. And the hippocampus is sending output to many different parts of the brain. So we really shouldn't think of the hippocampus as doing anything on its own. It's part of a much, much larger system, which is the nervous system as a whole. Okay, so we started studying the diversity of neurons in the hippocampus. And we used a method called RNA-seq, which allows us to determine all the genes that are expressed in any particular cell. And we've done several studies using this method, and we've created this database called the Hippocampus RNA-seq Atlas, or hipposeq. And there are a number of different data sets in this website that you can go to and explore the data. And I just want to show you that in this one study, Mark Sombrowski found that in an analysis of single cells and gene expression in single cells, that in this plot here, which is called the t-SNE plot, it looks almost exactly like a map of the globe. India is missing, it seems. But each of these little clusters of continents of cells are different cell types. And we were able to map these clusters of gene expression onto physical locations in this expansive structure that we call the hippocampus. And so we've learned that there's tremendous diversity in this cell types in this structure projecting out of the hippocampus to different parts of the brain. We also have identified diversity across the different regions of the hippocampus. So for example, dentate gyrus, CA3, CA1, dorsal and ventral, and several other aspects of the diverse collection of cell types have been probed in depth using this approach called RNA-seq. And I don't want to talk about this in detail today. I'll talk about it a little bit later on, but I did want to tell you about this resource. Okay, so I've told you that there's a diverse collection of neurons in the brain, including in the hippocampus. They observe complex cellular functions. They have complex connections. And what I didn't mention is that we should also not forget that there are also astrocytes in the brain. That's another important cellular component of the brain. And we have recently published a paper showing that neurons and astrocytes interact in meaningful ways that may mediate some signal transduction and information processing as well. Okay, so that is a sort of fairly long-winded introduction into how I view the complexity of the brain. Now let's talk about real data on forming memories and using memories. So we're going to talk a lot about the hippocampus, and to put you into the right context, I want you all to think about where you are right now, okay, and how you know where you are, and where you're going to be going next at the end of this seminar, and how you're going to get there. Now, there are a lot of ways that you can know where you are. You just look around the room, you take in a few visual cues, and then you make a comparison of those visual cues to some map of some memories of having been in this room before. And you say, oh, yeah, I'm in the seminar room at Georgia Tech. You could even close your eyes, and you could still know where you are. So this is an interesting cognitive function that the brain is able to tell you where you are, even in the absence of any ongoing sensory information. And you're able to imagine, you're able to do mental time travel and think about where you'll be going next, and even imagine the path that you'll follow to get there. Now, these are functions that the hippocampus is thought to serve. And in the next two sections of the talk, I'll provide some data that I think will not solve this problem of how this works, obviously, but touch on these issues. So I just wanted to get you thinking about this issue of how you know where you are, how you know where you're going. So obviously, memory is a very important part of that. We have to remember locations where we've been in the past and compare them to the inputs that we're getting now. The hippocampus is known to be involved in this. And since we're going to be talking about the hippocampus extensively, I want to talk about the circuitry a little bit. So you've got oh, this thing's not going to work. I'm going to use the laser pointer. Over here, the dentate gyrus sends its projections to CA3. CA3 sends its projections to CA1. And CA1 sends its projections to the subiculum. There's also entorhinal cortex, which serves as both an input to the hippocampus and an output of the hippocampus. So this is a well-known structure of the hippocampus, and I want to sort of schematize it for you in the following way. So we can think of this as a big loop. The entorhinal cortex provides input to the dentate gyrus, then that goes to CA3, to CA1, to subiculum, and back to the entorhinal cortex. However, entorhinal cortex also provides direct projections to CA3, and to CA1, and to subiculum, and CA3 has recurrent connections extensively within it. So there's a recurrent theme here, which is there are many, many loops in this system. There's this one big loop that I talked about. We can think about a slightly smaller loop here, or a smaller loop here, or even a smaller loop here. Now, to add further complexity to this, there are different cell types and different pathways in the subiculum. And I'm going to talk about Mark Zimbrowski's work to try to uncover the cellular basis of these different pathways and ultimately their function. And he did this with help from a lot of people. This work is now published as of last year. So I'm going to go through this fairly quickly. So one thing that we know is that there are many, many outputs of the hippocampus. There are many different structures that receive input from the hippocampus. And we also know that these are divided into different pathways. So this is work from Jim Knierim's lab, as well as many others, including our lab, showing that there are two populations of cells in CA1 and two populations of cells in the subiculum that form these two distinct loops here. And I'll refer to them when we'll be focusing on subiculum, so I'll talk about distal subiculum and proximal subiculum. And you'll see that proximal CA1 actually projects to distal subiculum, and distal CA1 projects to proximal subiculum. And they're interconnected with medial and lateral entorhinal cortex in the ways indicated by the colors here. and they have different downstream targets as indicated by these colors, which we'll take advantage of. Okay, I'm going to show you a bunch of pictures of the subiculum, and they're going to be in coronal section here. We're going to be zoomed in on the subiculum here, and the subiculum is this region right here. This is CA1, this is subiculum. Proximal is the subiculum closest to CA1, distal is furthest away. And so what Mark began by doing is showing that in the mouse, as the circuitry had been determined in other species like the rat, there's indeed this projection-specific architecture. And he did this by injecting retrograde beads into different structures. So here he's injected into, sorry, these are retrograde virus injections in retrosplenial cortex, ventral hypothalamus, or medial entorhinal cortex. And you see the labeling in subiculum here is restricted to the distal portions of subiculum. And you can see this best because there's, with this other injection of a different colored tracer in the nucleus accumbens, which labels the proximal part of subiculum. So if you superimpose these two, you can see clear distinction between the distal cells, which project to these three regions, and the proximal cells, which are projecting to nucleus accumbens, as well as other targets that we haven't labeled here. Okay? So we showed in this work that there's a clear distinction, two separate sets of cells projecting to different areas outside of the hippocampus and spatially localized to different parts of the subiculum. Here's another example of retrograde labels to prefrontal cortex and lateral entorhinal cortex, and you can see that they co-localize with this nucleus accumbens projection. And as always, there's one exception. There's a small population of cells that project to the thalamus, and they sit right at the border between proximal and distal subiculum. Okay, so we have these, we're going to focus on these two populations of cells which are projecting to these different downstream targets. And what we wanted to do, we've shown that they have different outputs. In other work, we showed that they have different inputs and that they have different firing phenotypes as well. But what we really wanted to do was get genetic access to these two populations of cells so that we could turn them on or off and determine the effects on the behavior of the animal, memory-guided behavior. And we were able to do this by injecting viruses into these targets. Now, now this is not viruses, this is beads. And then we use these different colored bead labeling to sort out the cells. So we dissociate the cells, and we sort them out, and we do RNA-seq. And the idea here is to determine gene expression in these two populations of cells, which we've done. And I told you about the website where you can find all the data. And we find that indeed, in these two populations of cells, there are many genes that are differentially expressed. So if we plot the expression of genes from one group of cells against the expression from another group of cells, all these colored dots represent genes that are differentially expressed. There's hundreds of them in each case. And if we look at what kinds of genes are differentially expressed, we find that many of them have functions that we associate with the functions of neurons, things like axon guidance and adhesion molecules, peptides and receptors, synaptic function, voltage-gated channels, et cetera. And you can see by these colors and these two different populations of cells that these genes are expressed very, very differently in these two populations of cells. So they're different cell types, they're expressing different genes, they're sitting in different places, they're sending their output to different places, they're getting input from different places, and so on. But we did all of this so that, and we validated that these differentially expressed genes using RNA-scope. I'm going to skip over this because of time. And we've also done this with single-cell RNA-seq. I talked to you a little bit about this before, and we find indeed that these distal and now each point on the graph here, instead of being a gene, this is a cell, and it's clustered into groups that represent the distal population, and the proximal population actually appears to be at least two different cell types. But we're going to lump them together and treat them as one. Okay, so we have now different gene expression in these two populations. And what I told you we wanted to do is take advantage of this differential gene expression to turn the cells off and determine their effects on behavior. So here's how we do this. We found Cree lines for these differentially expressed genes. We determined that that Cree is indeed expressed differentially in these two populations of cells. So now we can use this Cree line, and we can do an injection of a virus, in this case, into proximal subiculum. And it's carrying a label, M. cherry. So the virus is only expressed in Cre-expressing cells in this Cre line, and therefore the population of cells expressing this transgene is restricted to proximal subiculum. And you can see this because we inject another retrograde tracer expressing GFP in the distal subiculum, and you see these are non-overlapping. And they're not, you know, it's not that this is restricted to proximal subiculum because that's where we put the virus. For sure, the virus can spread over to more distal subiculum. It's restricted because of the requirement for the Cre for the expression of the reporter gene, which has its M-cherry label. We can also do the opposite experiment using a different Cre line, which is selected for distal subiculum. We inject the virus for M-cherry now in the distal subiculum. You can see that here. The GFP label is a retrograde tracer that goes to proximal subiculum. Again, non-overlapping projections. Okay, so now we assess behavior. And we're going to use, what you may have noticed, if you were looking closely, is that we didn't just express M-cherry in these cells, we also expressed HM4D. HM4D is a dread receptor, which can be activated by a ligand to silence the neurons. And we showed that this works really, really well in subiculum. So we're going to assess behavior while silencing one or the other population of cells. And we did several different tests of different behaviors and found no effect. But then, interestingly, we found an effect on a test called the Morris water maze. Now, probably many of you are familiar with the Morris water maze, but for those of you who aren't, here's a video. Come on, play. The mouse is put into an opaque pool of water. They're good swimmers, but they don't really like being in water, so they're looking for a way out. And they can't climb out of this pool, so they just swim around, desperately hoping for some escape. And indeed, there is a platform hidden in the water somewhere below the surface of the water. And the mouse eventually will find this platform. And once he finds it, he stands there on the platform and just stays there. But initially, this is just a search, a random search, until he finds the platform. Okay, but now if you repeatedly put him back into the same pool of water, he'll learn the location of the platform. This is now a different animal. But this guy's already been trained, and he knows exactly where the platform is, and he swims straight to it. Okay, so here's the experimental design. Every day, the animal is exposed to the platform multiple times, and every day, the platform location is changed. So it takes a few trials on one day before the animal learns the location of the platform, and then we're mean, and the next day, we switch the location of the platform, and they have to learn a new location all over again. Okay, then we do this 9 days in a row, and then on day 10, we inject either CNO or vehicle. We do the same type of training procedure, but then on one extra trial, we do what's called a probe trial, where the platform is not there. And you determine whether the mouse has learned or remembered the location of the platform by whether it looks for the platform in the right place. And then on day 11, we do this again with either the vehicle or the CNO, depending on what we did on day 10. Okay, so here are the results for the control. You can see when there's a probe trial, the animal swims most of the time in the location where it's expecting to find a platform. Okay, so they've learned that on this day, the location of the platform is right here. They swim around there most of the time. When we silence the proximal subiculum, it has no effect. But when we silence the distal subiculum, the animal shows no sign of remembering where the platform is located. So it doesn't learn the location or remember the location of the platform when we've silenced that population of cells in the distal subiculum. And here's the group data to show you this is true over many, many mice. Okay, so you actually can learn a little bit more than this from these experiments because of an interesting observation that when you move the platform on the next day, the animal seems to remember the location from the previous day. And the reason we know that is that even though there's no platform here where it was the previous day, on the first trial of the next day, the animal will spend most of its time swimming in the area of the platform from the previous day. So the animal clearly remembers where it was yesterday and eventually realizes it's not there anymore and learns the new location of the platform. So there's an interesting thing here, which is you can test whether the animal, when we silence distal subiculum, has a retrieval deficit or an encoding deficit. So if it's a retrieval impairment, what that means is on the CNO day, the animal shouldn't remember what happened on the previous day. But if it's an encoding impairment, then on the following day, when the CNO has worn off and the cells are no longer silenced, the animal should show no sign of having learned it on the previous day. And what we find is that it's actually very consistent, that the retrieval is intact, and it's consistent with an encoding impairment. Okay? So even though we've silenced distal subiculum and the animal can't learn a new location of the platform, it does show signs of remembering where the platform was on the previous day. Now, here's the group data. This result was very surprising to me for the following reason. The subiculum we think of as the major output of the hippocampus. And we think of the hippocampus as the structure that forms the memory. And so my prediction was that, well, if we silence the output of the hippocampus, the memories can still be formed. All this circuitry in CA1 and CA3 and the dentate gyrus, that's all still intact. The memory can be formed. But if you can't send that information out to these other areas of the brain, the animal won't be able to use that information. But in fact, it's the opposite, that the information going out of the subiculum needs to be there in order for the animal to form the memory. This is a very surprising result. Now, one possibility is that we've disrupted this loop, or one of these loops that I talked about before, by cutting the output right here. So maybe cutting the output here was less important than cutting the output here. And these are the kinds of things we can test in follow-up experiments. But the major hypothesis I have at this point is that some kind of long-range loop has to be intact in order for the memory to be formed. That this long-range loop, for example, if you cut it anywhere, it's not going to be able to form a memory. And again, this is a somewhat surprising result that changes, for me, the way I think about how memories are formed. They're not just formed in the hippocampus. They're formed in the hippocampus and other circuitry that's associated with output from the hippocampus to other parts of the brain. Question, Joe? So yeah, Joe's question was whether the Cree lines were specific to dorsal versus ventral subiculum. And the answer is that there's some expression in ventral subiculum as well. There's also expression in other parts of the brain. But what we've done is by doing virus injections, we restrict the region where the DREAD receptors are expressed. So most of the DREAD receptors are going to be expressed in dorsal subiculum because that's where we inject the viruses. So it's an intersectional approach that allows us to restrict the manipulation. Okay, where are we now? This thing keeps exiting on me, not sure why, doesn't matter. Okay, I talked about how we can cut, potentially in future experiments, cut in different places. Okay, I've talked about what we're trying to do overall, formation of memories. Let's talk about using memories. Okay, so back to this question. How do you know where you are? You have to use memories to know where you are. I'm going to talk about this work that was done by a postdoc, Xinyu Xiao, and this is part of a collaboration with Jeff McGee's lab. And Ying-Shui Wang was a postdoc at Jeff McGee's lab, and she contributed also. Okay, so again, hippocampus is involved in spatial memory. And we want to try to understand how the hippocampus knows where we are. So we're going to record from neurons in the hippocampus, specifically in the CA1 region of the hippocampus, while an animal navigates a virtual environment while running on a ball. The environment looks like this. It's an oval track. There are 6 discrete locations. The animal runs clockwise in this virtual track. And there are different cues on the wall at each position. So I'll refer to these as locations 1 to 6 and cues A through F. And we'll move the cues around a little bit. Here's a weird sort of hybridized video that gives you a sense of the animal running. This video is not working, but the animal runs on the ball. That controls movement in this virtual space through this artificial environment. So while the animal's doing this, we can put an electrode in, do a whole cell recording in CA1, And CA1 cells, as I mentioned, get input from CA3 as well as from the entorhinal cortex. And you can see a beautiful labeling here. This work by another postdoc, Eric Bloss. This is labeling inputs from the entorhinal cortex, which are restricted to the distal tuft dendrites, and the rest of the dendrites get their input from CA3. So we're recording from these CA1 cells, and because we're doing whole cell recording, we can evaluate not only whether they're spiking or not, but also what kind of synaptic input they receive. So here's how the experiment works. The animal's in this virtual environment, we're recording, and in many cases, as the animal runs a lap, there's nothing. There's no firing. We don't see any synaptic input. And we actually take advantage of this. And we take advantage of a beautiful, we can show that this in fact is a real neuron. We can reconstruct it histologically later on. But it's silent. It's a silent cell. Now we take advantage of a beautiful trick that was discovered by Katie Bittner, a postdoc in Jeff McGee's lab. And this is called behavioral timescale synaptic plasticity. I think this is one of the most profoundly important discoveries of the last decade or so in the field of synaptic plasticity. And what Katie discovered is that sometimes neurons will be silent and then suddenly they'll form a place field. And when they do that, there's a plateau potential associated in the place field. And she could, in fact, create an artificial plateau potential and artificially create a new place field. So that's what we've done here. On the next lap, on lap number 3, we just inject a bunch of current into the cell while the animal's at location A. We do that a few times, and on all subsequent laps, the cell fires action potentials in that same location where we artificially injected current. Okay? And then in a follow-up paper, what Katie showed is that there's some beautiful synaptic plasticity that occurs as a result of this big plateau potential, and that's responsible for the formation of this place field in this CA1 cell. Okay, looking at this a little more closely, when the cell fires in the place field, there's a few things. There's spiking, of course. There's a ramp of depolarization, which has been filtered out here. And there are theta oscillations that increase in amplitude when the animal is in the place field. Okay, so we can induce a place field. And one of the nice things about that is we have control of these cues. And we can ask, what is it about that location that is causing firing? So to do this, what we do is we duplicate the cue. We move a cue, or we duplicate a cue at location 1, which is an A, and we put it over here at location 4. And so first we induce a place field in this location, which is location 1. And then we duplicate it at location 4, and you can see it produces a little bit of firing. This is a little bit wimpy, but there's some firing. And across a population, the effect isn't that small, actually. It's reasonably significant. You can see a significant amount of depolarization at this location where we've duplicated the cue. The red is following the duplication. You can see there's depolarization that wasn't there in the control. There's also an increase in theta amplitude and some spiking. Okay, so there's partial induction of a place field just by moving the cue. So it seems like the cue, the visual cue, is a really important piece of information that the animal uses to produce its map of where it is in the hippocampus. Now we thought maybe one of the reasons it's only partial is because when the animal is actually at location 1, it can see forward to location 2 where there's a different QB. Then when we duplicate QA at location 4, it sees forward to location 5 where there's a QE. So it's seeing different things. Let's now change the situation. It's a virtual environment, so we can do this however we want. And we put a virtual curtain here, which blocks the animal from seeing B when it's at location 1. It only sees A. And it blocks it from seeing E when it's at location 4. It can only see whatever it is at location 4. And when we duplicate the cue in this way, we get a much more striking effect. There's pretty much full-blown induction of place field firing at the location of the duplication. You can see this in the group data over here. So this is evidence that the only thing that's really driving place cell firing in this case is the visual input that the animal receives. And if we control the visual input so that it's only QA, then we can put QA anywhere in this environment and it will drive firing. So that means that the QA is, this visual cue is sufficient to drive firing of these cells. Now, if we, instead of duplicating QA, we move it from location 1 to location 4, that moves the location of the place field firing, okay? It's gone at the original location. You can see that in the summary data over here. So what that means is that not only is that cue, that visual cue, sufficient to drive the firing of the cell, it's also necessary. So without it, you get nothing. Okay, so we have perfect control over what's causing this place cell to fire. And that's pretty surprising in a way, because what it means is that the history of what the animal saw before it encounters A doesn't really matter. The fact that it saw D, then E, then F, then A is no different than seeing B, then C, then A. Okay? It has no recent history effect on the place field firing. That's a little bit surprising. But we knew that the representation of environments, or of cues in environments, is very context dependent in the hippocampus. So we asked, what happens if we take this cue A and present it to the animal in a totally different context, a completely different environment? So the animal learns to run in two different environments, an oval track and now a triangular track. And we have one common cue in the triangular track, that's cue A, which we've been talking about, is also present in the triangular track. And we can move the animal in virtual reality from one location to the other pretty much instantaneously. So what happens, call this context 1 and context 2, what happens if we induce a place field at position 1 with cue A in the first context, and then move the animal into a new context? We introduce the animal into the new context at position Y, and then from the very first time it sees A, in this new context, it does nothing, okay? So previously, you might have looked at that firing of that cell and said, well, wow, that looks like a cell in the visual cortex. It's just responding to a visual stimulus. But this is very different from a cell in the visual cortex because it's completely gated by context. And not only is it not firing, there is no depolarization whatsoever. Okay, so somehow context is an almost perfect gate of the representation of this object in the animal's environment. Here's another example where when we put it into the second context, there's no place field firing associated with QA, but we have place field firing associated with a different Q, X. And the summary data are shown here. Here's the control ramp depolarization in the original place field in the original context. Absolutely nothing when you present it in the new context. How are we doing on time here? We're going till 12:15 or 12:10, right? Something like that? Okay, good. All right, so how does this work? The simplest way that you can imagine that a cell could encode a sensory stimulus and a context is that it receives some synaptic input associated with each of them. You sum it. This goes above threshold for firing, and you get action potential firing. This is clearly not how it works. And the reason we know this is not how it works is because when we changed the context, there was no leftover representation of the sensory input. The gating is occurring in some other way. And the most likely place to think about where that gating occurs is upstream in the CA3 region, which provides the majority of excitatory synaptic drive to these cells. So here's what a CA3 neuron looks like, by the way, from this mouse light project. This is so cool, I had to show it. This is a reconstruction of the axon arborization of a single CA3 cell. And this neuron has about 10 centimeters of axon associated with a single cell. And it was reconstructed by the mouse light team in 8 hours. Previous reconstructions of CA3 cells were taking months prior to the work of this team. So that's what these CA3 cells look like. They're providing massive amounts of input to CA1, so it's a logical place to now go and look for this context-dependent gating. Okay, so we do the same kind of experiment. Now, there's a difference here, an important difference, which is we did these experiments using extracellular recording. The whole cell recording is harder in CA3, so we started with extracellular recording using silicon probes. That means you can't induce a place field, so you have to rely on cells that already have a place field, and you don't know quite as well what's driving them, but you can infer it. So here's an example. Here's a cell that's responding right around the end of QA, beginning of QB type of area, okay? So now we duplicate QA at a new location. We shift it, we don't duplicate it, and that moves the place field firing to the new location. So just like in CA1, it seems like the cell is responding very reliably to the visual input. Here's another cell where it's similarly pretty good. Here's the visual input that's driving this cell is probably around A and probably a B here as well. And when we move the A to the new location, leaving a blank here at position 1, we clearly induce place field firing at the new location. There's some leftover firing at the original location, probably because QB was also driving the spiking of this cell. So again, very consistent with the notion that visual input is a very important driver of these cells. Here's another example where we shift the natural place field occurs at location 4, probably largely driven by QD. So now we move QD from position 4 to position 1, and the firing moves close to that same location. There's a little more variability and a little more noise in CA3 than in CA1. Here's an example when we moved the location of the place field, there's some leftover firing at the original location, so it's not quite as clean as what we saw in CA1, but nevertheless, this is a pretty robust effect that when we move the cues, the place field firing gets shifted from the old location to the new location. You can see there's some leftover firing at the original location, but not much. Okay, so what about context? Context also seems to gate the firing. So we have driving of the firing by the visual input in CA3. What about this contextual gating? We do the same contextual gating experiment. This is a little bit messy. This one, the cell has a lot of firing at its original place field. We present the same cue in a new context, and there's a little bit left, but not much. The other cells are cleaner. This one has complete gating of its place field firing in response to context. This one also nearly complete gating, and this one complete gating. Okay? So in CA3, the situation looks very similar to what we saw in CA1. Now, what have I told you? We've got sensory-driven place fields in CA1. They're strongly gated by context, and the same thing is true in CA3. I told you that this model of contextual gating of the cell's response could not be true in CA1, but what about in CA3? Oh, sorry, instead, what we think is happening is in one context, you get a population of CA3 cells are acting as input to CA1, producing these depolarizations in CA1, which sum to drive the spiking of the cell. Okay, so the gating, the contextual gating, is either occurring in CA3 or even upstream of CA3. By the way, I just want to briefly mention this. I probably don't really have time to go into it detail. This is published last year, a beautiful project done by another postdoc, Qinglong Xu, showing that these CA1 place cell responses are very voltage dependent. So they're an extremely nonlinear response to the synaptic input. That means that a very small, subtle change in synaptic input, which is going from the red to the black here, produces a big change in the response of the cell in its place field in vivo. And he did a bunch of in vitro experiments also to determine the cellular mechanisms and the ion channels that are responsible for this nonlinear amplification of place cell responses. And I think this is going to be very relevant to the mechanisms of this contextual gating. Okay, so I told you this model could not be true in CA1. It could be true in CA3, but based on the spiking data, it seems very unlikely that this is true because there's already really strong contextual gating of spiking in CA3. So instead, I want to present a model. This is a cartoon, back of the napkin type of model of what I think might be going on. And it provides sort of the basis for what we hope will become a computational model that can produce some testable predictions down the road. So the idea is that in CA3, you have a strong sensory response and a very strong sensory input and a small contextual input. These two things serve as input to a CA3 cell. They sum nonlinearly to produce spiking. The same is true in some other cells in CA3. And because these are all connected to each other by these recurrent collaterals, this population of cells reinforces its own firing, and you get a population of cells associated with a particular sensory cue in a particular context or environment. Now this population of cells that's firing in CA3 serves as input to CA1. Maybe there's a little bit of contextual input in CA1, but this has to be now enough to gate this firing in CA1. So really what matters in CA1 is what population of cells was activated upstream in CA3. Now to illustrate this for you, imagine that we put, so the way I'm describing this is a mechanism that involves cascading nonlinearities in a circuit. There are nonlinearities at several stages of circuit processing that can result in this very strong contextual gating that we see in CA1 and to some degree also in CA3. Okay, so now imagine what happens when we put the same sensory stimulus in a different context. This small change in the contextual input to a cell, let's say maybe it's a CA3 cell, can actually cause its spiking to be reduced or even to fall below threshold for spiking. And therefore, a different population of cells will be firing in response to this sensory cue in this context. This population that responded in context 1 will not respond in context 2 because that little bit of input from the contextual signal was enough to cause a large number of cells to fall below threshold for spiking. Therefore, the input to CA3 cells will be a little bit from this guy because he fired one spike, nothing from this guy, and nothing from context, and you have almost nothing left in CA1. So this concept of cascading nonlinearities, I think, is a nice model for how you might get something like contextual gating of sensory inputs. Now, the reason I asked the question, how do we know where we are? We have really, in these data, a neural signature that the animal probably knows where it is as soon as you change the context. That there is a very strong neural representation that the response to something we know drives the firing in one context doesn't do anything in another context. So it's a neural signature of that context, of that knowledge, if you will, of where the animal is in its environment. Okay, I'm going to stop there. Again, I want to thank many, many people at Genelia, including the people in my lab, all these other people that I mentioned. And I just would like to tell all of you that are considering for your future where to go, that Genelia is a great place to think about coming and doing a postdoc or even PhD work if you're at that stage. So thanks, I'll stop there, and maybe there's a few minutes for questions.
We have a few minutes for questions. If people need to duck out, go ahead and duck out. But if you have questions, raise your hand.
So really awesome stuff. I was wondering about the last set of slides, though. To what extent do you think the context effect could actually be driven by local geometry? Because I notice, especially when you do the virtual curtain situation. So when you take away, say, the cue for what the next location is, all the rodent has available to it is this sort of, just come around the corner here. And that's perfectly mirrored on the other side of the track. That is not the case when you move it into the triangle. So
yeah. So the question really is, what information is the animal using to determine which of the two contexts it's in, right? And there are two important differences. The first one is that in the second context, there's a novel cue. We labeled it Y on those slides, which the animal never sees in the first context. So in principle, that alone is enough information for the animal. It's experienced both of those environments before, during training. So in principle, that just seeing Y is enough to know, yeah, I'm in a different place now. There's also a different turn angle, as you point out, which the animal experiences before it encounters the relevant cue. So we don't know how important that turn angle is. So in future experiments, we want to take that out of the equation, either by creating two environments, both in oval tracks, or maybe even trying to create just linear track environments. So you can just have an animal running in repeatedly occurring sequence of cues that mimic the sense of running around in a circle. Instead, it's an infinitely linear track. And then you can take turn angle out of the equation altogether. So we're going to do some experiments like that. My prediction is that probably that initial cue Y is all the animal needs to know that, oh yeah, this is this other environment. But it's not 100% clear that that'll be true.
Right. Have you thought about in the oval track just shifting the cue one step forward? So on the other side of the curtain, basically. So now they're facing a turn. And so clearly,
Yeah, I
that's
mean,
really different.
we haven't done that experiment, but I would make a very strong prediction there that if we move it on the other side, the relevant cue to the other side of the virtual curtain, it'll produce partial firing because they can see past the relevant cue and they can see other things. So now their visual input is different. Yeah. And the part of the difference in their visual input is the turn angle coming up. Yeah. But all this stuff is, you know, easily addressable future experiments because we have full control over the virtual environment. Annabelle.
I think my questions might be kind of similar to Thackeray's, but specifically you said based on the whole cell patch experiments you had in CA1, you don't think history really matters to the cell. But then your results in CA3 were a little bit different, and I wasn't sure if you thought that's because you're doing extracellular versus patch and you have a little bit less control, or if you think that history might matter in CA3.
The question is whether history matters more in CA3 than CA1. I just want to point out that history you know, has different time scales, right? And what we showed is that the recent history doesn't matter. That is, if QA is the relevant visual cue driving the firing, it doesn't matter if it was immediately preceded by a B or a C or whatever. QA is going to work on it nonetheless. The longer history actually does matter, and that's the contextual gating, because the animal's knowledge of where it is, which it also formed by history and by memory, is relevant. But your question, I think, is about the very recent history, the cue it saw right before the cue that drives the firing, and whether that matters more in CA3 than in CA1. And we don't have any evidence that it does matter more, but you're correct in pointing out that it's harder to interpret the extracellular recordings than the intracellular. And the intracellular is beautiful because you can see whether there's any input associated with a cue in this particular context. In the extracellular, you can only tell if the cell is spiking. So Jeff McGee's lab is doing whole cell recording in CA3. This was a collaboration between our lab and his, so he'll hopefully have an answer to that question within a few months. But for now, I would say we don't know, but I doubt it. That's my guess. The reason I say I doubt it is because I think whatever happens, whatever the animal uses in CA3, that information is available to CA1. And you would see some signature of that, I think. Yeah. So the model is just the drawings that you that I showed you there. That's all it is right now. So we haven't assessed how dependent this model would be on the relative strength of sensory versus contextual inputs. I suspect that strong contextual inputs would also work quite well. What I was trying to convey by showing the small contextual input is that the point is that nonlinear amplification can accentuate small differences. And it's been shown in many past experiments that animals don't need much information, sensory information, to distinguish between two environments. And that's probably a consequence of pattern separation, that you show them two things that are subtly different and pushes neural circuits into different activity states, or different activity patterns, I should say, based on some small difference in the contextual input. So that's what I was trying to convey, but I would say it only gets easier as the contextual differences are greater and the contextual signals therefore increase. Oh, I would think so, yes. Yeah. Yeah, question.
Did you consider the possibility of temporal coupling, where, for example, the animal anticipated that they would see a cue, could that have an effect on the model as well?
So
Start them at different locations in context as well.
the question is about the animal's anticipation of a cue, and I think what we've shown is that animals although they may be anticipating a cue, there's no place field firing or place field response that is indicative of that anticipation. So even though the animals learned that every time I come around past QF, I'm going to see QA at this location, as soon as we remove that QA, not only is the firing gone, the synaptic depolarization is completely gone. So that would suggest that anticipation, there's no signal of anticipation that's visible in the hippocampus, at least in the way that we've assessed it. Yeah.
Having some fun thinking about nonlinear amplification not being static, and that as memories are formed, that you might lose that
Yeah, so there's two issues here. The question was about nonlinear amplification not being static, and that as memories are formed, you might lose that high gain. There's two issues here because we are looking at the hippocampus after memories have already been formed. We have a series of training trials where the animal is exposed to these environments to become familiar with and comfortable with the idea of running on this ball to change the virtual world. And so we're looking at it after memories have already formed. While memories are being formed, that is, the very first time you put an animal into a new environment, presumably there's a whole different set of things that happen to allow memories to be formed. One of them is probably this behavioral time scale synaptic plasticity that I mentioned before. That's also dependent on nonlinearities in the neurons. So there are different kinds of nonlinearities that could be invoked. There's also neuromodulatory transmitters and receptors like dopamine and acetylcholine and serotonin that can change the state of the system, that could in principle turn these nonlinearities up or down, turn the gain up or down. That could
regulate the bistable fire.
That's right. So they could regulate how readily the system is prepared to be modulated, to be subject to the nonlinearities that will form new memories versus the modulatory circumstances that may more readily produce recall, or the nonlinearities that are associated with recall of previously formed memories, for instance. So there's a whole host of nonlinearities in the system that can be all regulated up and down probably.